Hello, I'm Dr. 𝗫𝗕𝗃𝗏𝗃. I'm just wondering, am I audible to everyone? There are no sounds, so just checking the sounds. Am I audible?
Yes, you were
Uh,
audible.
good evening, everybody. I hope uh, you're going well. A warm welcome to the International Congress. I would like to invite Ms. 𝗕𝗂𝗀𝗃𝗏𝗏 have a floor for you. You can continue. Thank you.
Thank you, sir, for your warm invitation. So, Assalamu alaikum and a very warm good afternoon or evening to all respected professors, distinguished speakers, researchers, scientists, and academician guests and participants joining us from different countries around the world. It is a great honor and pleasure to welcome you all to the International Congress on Advanced and Sustainable Plant Science, Agrobiodiversity, and Human Health. I'm Nupur Akhtar, Head of Collaboration at Vitanova International Alliance for Sciences, and I will have the privilege of hosting today's Congress. Today's Congress serves as a global scientific platform decided to promoting research and innovations, sustainable plant sciences and agrobiodiversity, biotechnology and human health, international scientific collaboration, and knowledge exchange. This Congress is proudly organized by Vitanova International Alliance for Sciences and in a collaboration with Scientific Research Institute of Fruit and Tree Growing, Ministry of Agriculture, Azerbaijan, University of Swat, Pakistan, Shirt University, Turkey, Federal University, 𝗤𝗏𝗃𝗏, Nigeria. So, before we begin the research scientific sessions, it is my privilege to acknowledge our respected organizing committee members whose dedication and guidance made this international congress possible. So, the first one we have is Professor Dr. 𝗬𝗎𝗓𝗍𝒐𝒐𝒍𝒐, Professor, Department of Molecular Biology and Medicine, Federal University, 𝗤𝗏𝗃𝗏𝗏. Dr. Ilnara Kanegishi-Yeva-Ahmadova, HOD, Molecular Biology, Scientific Research Institute of Fruit and Tea Growing, Azerbaijan. Engineer Professor Dr. Madad Ali Shah, Vice Chancellor, Shahid Benazir Bhutto University, Sindh, Pakistan. Mr. Ghulam Murtaza, CEO and President of VNAS, Center of Agriculture, Biochemistry, and Biotechnology. Dr. 𝗤𝗃𝗕𝗓𝗏, HOD, Biotechnology, the University of Faisalabad, Pakistan. I would like to invite Dr. 𝗤𝗃𝗕𝗓 to welcome our participants as well as our guests with your few words. Thank you. Dr. 𝗤𝗃𝗕𝗓, ma'am, are you there?
Yes, thank you very much for introducing all the organizing committee and all our participants today. I would like to welcome you all to our this international congress, and definitely it is need of the day to talk about some green techniques, some sustainable approaches that address directly, indirectly to some of the processes or the practices we are actually implementing in industries, in any capacity, in any institutes. We need to revive all those processes and advance by implementing some innovative techniques that we need today. And this is basically the theme to collect the knowledge or the advanced research, innovative techniques that are being experimented with. And definitely our guest speakers will talk about their latest research that definitely address some sustainable approaches and green processes, which are definitely environmental friendly, cost effective. And yes, we have lined up a very good team of some international speakers with us today. And such platforms are very important. So I would like to thank the conference chair, Vita Nova, for taking this opportunity to provide all of us this important platform where we can around the globe sit together and talk about the research and the advancements that is happening to address these global pressing issues. So without taking more time, I would like to welcome you all again. And with this, I will pass on to the organizing committee or the guest speakers if they are here. So thank you very much for opening remarks. I appreciate that. Thank you.
You are most welcome, ma'am. And the last one is Dr. Shahnur Khan, Director IASHS, CEO, International Center for Plastic Pollution Control, USTB, Pakistan. Now, for another remark, I would like to call Ms. 𝗤𝗃𝗕𝗓𝗏, Vice President, Vitanova International Alliance for Sciences. Ma'am, kindly take the floor and welcome the participants as well as the guests with two or three sentences. Thank you.
Assalamu alaikum everyone and a warm greetings. Here is me, Hafsa Sarfraz, an MS scholar in plant breeding and genetics from University of Agriculture, Faisalabad. And currently I am serving as Vice President at Vitanova International Alliance for Sciences. It's really pleasure to see all the international speakers here, and I really warmly welcome all the participants to the International Congress, and best wishes for all. Thank you so much.
You are most welcome as well. Now, the rest of them are Dr. Ayman Al Shabak, Assistant Professor, Department of Field Crop, Shirt University, Turkey. Dr. Wasir Ali Metlo, Associate Professor, Shahid Venzir Bhutto University, Sindh, Pakistan. And Dr. Lawan Hassan Adamo, Associate Professor, Federal University, Dutse, Nigeria. If our respected sir is present, Dr. Sir Mr. Ghulam Murtaza, the CEO and President of Vitanova International Alliance for Sciences, kindly take the floor and welcome our respected speakers as well as the participants so that we can move into the main topic. Thank you.
Thank you so much, Ms. Nopokto, for inviting me. I really warm welcome all of you our respected guest speakers and respected organizing team members from representatives from four countries: Nigeria, Azerbaijan, Pakistan, Canada, and from the Turkey. So moving forward to it, uh, now we will move forward towards the our main topic. So here in our hosting team right now join us Dr. Kenal from Scientific Research Institute 𝗤𝗏𝗿𝗀𝗃, Azerbaijan, along with Dr. 𝗤𝗏𝗿𝗀𝗃𝗃 from 𝗤𝗹𝗵𝗃 University from Turkey, and from the Nigeria here join us Dr. Ibrahim Ismail Saleh. So I really warm welcome all of you in today's International Congress. We will move forward towards our official announcements of our guest speakers.
Now we will move to our main topic and invite our speakers for their valuable presentation. First of all,
जी
it
जी.
is now my immense pleasure and honor to
Am
welcome
I audible?
our.
Am I audible?
No, sir, you are not audible. Now you are audible.
Okay. So we
So,
will
can I
move forward
start?
towards. Yeah, we'll move forward towards our presentation. This one, please invite. Yeah,
Sir, can you repeat that?
we will move forward with our this presentation. Professor Kazipan, you can continue. Please invite them.
Okay, good
Nupurta, you can.
afternoon.
Nupurta, please invite Dr. Professor Kazipan. Yes, yeah.
Okay, good afternoon, distinguished professors, respected participants, and dear colleagues. It is great honor and pleasure for me to be here today. Thank you very much for your kind invitation. And also, I show you my presentation. I tried, sorry.
Thank you, Miss. You can start your presentation. We would like to invite you as a speaker. The floor is yours.
I would like to share my screen. Can I share my screen also?
Actually, screen is shared. You can start the presentation.
Hello, dear Dr. Professor Kazipan. Is our
Please wait a moment. Please wait a moment.
okay?
I
Okay.
shared my screen. I want to try.
Okay, there is a below option. You can share screen from here. There is a below option, computer. You can see computer option below.
Yeah, yeah. I have
Yeah.
1 minute, please. Can you see my screen also? Okay, I can present using your shared screen because I will not share my screen also. Okay?
Your screen is visible. You can continue.
Okay. Sorry.
Yeah, yes.
You can see my presentation. Can you see,
Yeah.
dear professor?
Yeah.
Okay. But this is PDF, sorry. Okay. Okay. First of all, I would like to greet everyone and wish you a good day. I am a director of TNT Product Research and Application Center at 𝗔𝗴𝗿𝗍𝗿 University, which is located in Rize, Turkey's main tea producing region. Turkey has an important geographical position connecting Asia and Europe. It is also the largest tea consuming country. Tea has adapted very well to the sloping lands of Turkey. You can see image from different tea orchards in Turkey. And also our harvest season, these days, our harvest season, you see our tea producers while collecting tea. This is our university, and this is the different picture in Rize, the castle of Rize. As in other tea producing countries, tea in Turkey is an important source of income for its growers. In Turkey, black tea is the most widely produced and consumed type of tea. But in recent years, the production of different types, such as white, green, and oolong tea, has also begun. As seen in the figure, Turkey is one of the countries located at the northernmost 𝗕̌şi, where tea is cultivated. When we look at the table, we see that China is the largest tea producing country, while Turkey is the leading tea consuming country in the world. In Turkey, tea cultivation is carried out on approximately 8 to 1,000 hectares of land by around 205,000 producers. The annual production of fresh tea in Turkey is approximately 1,400,000 tons, while the production of processed dried tea is around 290,000 tons. The total number of the factories is 183. Rize province in Turkey is known as the capital of tea. In terms of total tea area in Turkey, Rize ranks first with 530,000 acres. Especially in studies conducted by FAO, when the world's tea producing countries are evaluated, several threats to the future of tea cultivation have been identified. These include, for example, the increase in extreme meteorological events, warmer and dry climate condition, reduction in water resources, increase in drought severity, deterioration of water and soil quality, and the others you can see. In response to potential Terites facing tea cultivation, several strategies have been identified, particularly under the leadership of 𝗏. Today, I will give a presentation on two important topics among these titles related to the studies carried out our university, the conservation of genetic resources, breeding, and the development of resistant variety. Before starting the breeding studies at our university, we collected tea genetic resources and established the tea gene pool. After 4 years of research, 2,034 tea genotypes were identified across all tea growing regions in Turkey. The genetic resources were collected in this region, Trabzon, Rize, Artvin, and Giresun provinces. During this process, we visited many field sites and collaborated with our tea growers. With these projects, we obtained many genotypes with very high difference characteristics. Here you can see our purple tea genotypes. We propagated these different genotypes and planted them in the National Tea Gene Pool in our university. Each genotypes were individually documented, and also all the collected data were entered in the computer-based system. Different morphological and phenological parameters were measured. All genotypes were also maintained in the greenhouse for presentation, also preservation. Each genotype was processed in the various tea types, and the quality of the produced tea tray was assessed. You can see our gene pool. We examined each genotypes according to 𝗏𝗏 criteria also. We described the flower and shoots and leaves also. You can see different tea types flowers. We identify all flowers characteristics and all different characteristics also. The growth and developmental performance of the genotypes evaluated our research team also, and we write a book entitled 𝗦𝗂𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀𝗀 scope of our projects, the genetic resources were conserved by surviving 4 provinces and 31 districts. Producing tea and development of the first long, white, and green tea culture in our country are among the major outcomes. In addition, within the scope of our projects, the molecular characterization of Turkey tea genetic resources was carried out. And also, a tissue culture propagation protocol for tea was developed to provide seedling to procedures. In collaboration with the Turkish Energy, Nuclear and Mineral Research Agency, we have started mutation breeding studies in tea. Dear professors, are you seeing my presentation good?
Yes,
Yes,
your
it's
presentation is visible.
Okay. I am 14 slides, okay?
yes,
Sure.
okay.
Okay, thank you. In our mutation breeding studies on the 𝗔𝗴𝗃 culture, we first determined the effective mutation dose and then applied this dose to 4,000 cuttings to perform our observation. No, but survival was detected at doses above 30 gray. The best rooting and shoot growth performance was observed at 10 gray mutation dose. And 11.405 gray cobalt-6 is the effective mutation dose in our genotypes. And following the application, single node leaf cuttings obtained from the irradiated shoots were planted in the rooting medium for propagation. You can see shoot and leaf color variation were observed among cutting taken from the same cultivar. Dear Professor, dear Murtaza said to me, please say the moderator moves the slide. My slide is 44. Is that okay? This slide. I shared your screen. I shared this slide.
Yeah, yeah.
Is it okay?
Yeah. Okay. Just let me
Okay.
know on which slide you are currently.
44.
Okay, 44.
I showed different slides. You can see?
Yeah, it's 44. Shoot and
Yeah.
leaf color variation.
Yeah.
Yeah, continue. Please have follow. Yeah, dear Professor Kezban, you can start. Yeah.
Okay. Here you can see different mutation plants.
Yeah, we are on 44 slide. Shoot and leaf color variation.
Miss Professor Kezivan, you are not audible. Kindly check your microphone, please.
Yeah, now please.
Okay, I am in here again.
Yeah,
Which
it's okay.
slides are ended? 40,
44. You can continue, please.
44. Okay, please be quiet, please. Sorry. Okay, I start 44 slides again. You can see the mutant plants, shoots, and leaf color variation were observed among the cutting taken from the same cultivar. This type is the same cultivar and added different mutation. Those. Sorry. Can you hear me?
Yes, we can hear you, Miss.
Okay, someone say to me, we don't hear you. Okay. I want to ask you.
Please go on.
Okay. We performed various measurement and observation, for example, 47 slides, observation and measurement in mutant plants such as shoot length, tea leaf and one bud length, leaf width, leaf length, chlorophyll content, and lateral shoots number and flower number also. After that, to identify the DNA sequencing variation among the mutant tea plants affected by gamma radiation, screening was conducted using 14 ISSR and 16 SRAP marker. And 49 slides, you can see differences between 𝗔𝗴𝗿 and its mutant were found to range from 48 to 94 according to the genetic similarity based on SRAP markers. The mutants number 139 and 203 were found to be most closely related with a genetic similarity rate of 96%. According to genetic similarity index based on SRAP markers, the differences between 𝗔𝗴𝗿 and its mutants range from 51% to 97%. This figure shows the most closely related and the most genetically distant mutants. And in these projects, mutation breeding projects, measurement and observation are currently being carried out in the second generation. And our climate resilient breeding studies, we have launched a new project to determine the drought tolerance of the cultivar candidates in our gene pool. 4 slides. And also, in these projects, 14 tea variety candidates from Turkey, 2 tea variety candidates from Japan, and 2 tea cultivar from Azerbaijan will be evaluated for their drought tolerance. The study aims to identify drought-tolerant variety and candidates varieties suitable for the cultivation. And you can see our plants subjected to drought tolerance testing and control water treatments. And you can see in these slides the stress treatment were applied to 1-year-old tea seedlings. The experiment were conducted under controlled greenhouse condition. At the same time, 58 slides, the genotypes grown in the field were also observed during July and August, and their responses under hot water condition were recorded. And 58-9 slides, you can see within the scope of the first work package, criteria such as leaf blade orientation and leaf color are being examined. Leaf blade orientation observation with our project advisor, Prof. Yoriki Nakamura, the director of the Tea Research Center, Suzuka University, Japan. And also live surface temperatures was measured using the infrared thermography. And then the work package 2, we determined of the morphological and phenological reaction. And the different work package 3, determination of metabolic differences that may occur in tea under drought stress, such as protein, catechin, caffeine, chlorophyll B, and carotenoid content, superoxide dismutase, catalase, and malondialdehyde content. In addition, 6th slide, you can see, in addition, a scale was developed based on the responses of tea plants under drought stress. 0 is no visible effects. The 1 is the leaves taking a position parallel to the surface. 2 is slight curling of the leaves. 3 is the marked downward curling of the leaves. 4 is the full downward swirling of all leaves. And 5, stem dropping and leaf drying. In the image of the culture on the 12 days after stress, you can see the 12 days after stress, our genotypes, and plants that weren't water for 24 days in here. And you can see resistant and sensitive plants in here. The study is currently ongoing. In the preliminary experiments, 6 drought tolerance cultivars have been identified. And the 67 slides, you can see our hybridization breeding research. We are currently conducted hybrid breeding studies in tea using both stress-tolerant and high-quality cultivars. Our main objective is to develop new variety that are biochemically rich and have superior inflation quality. Before the hybridization studies, the floral characteristic of the genotypes to be used in crossing were examined. We are crossing the different genotypes, and we are also examining the pollen viability and germination capacity of the genotypes. And 7.1 slide, you can see our hybridization studies have just started. One of our main goals is to develop tea culture with high information quality. We also organize training program on tea. One of our important activities is the training we organize on tea. We organize training on different work topics that I just explained. Experts from different countries also participated in our training program. And one of this training, our university and Çaykur organized a particle training. You can see the different organization, different training course, and tissue culture course. And also, to date, we also have visited different countries for international cooperation, and they also visited us. Here you see the team from Iran. We also made technical visit Lejan in Iran and Azerbaijan, and we have also been collaborating with Japan. Here you can see our visit to Kawasaki and Ochi companies in Suzuka tea region in Japan. And also we collaborated as Georgia, Pakistan, Sri Lanka. You can see our collaboration with Japan, Sri Lanka, Azerbaijan, and Pakistan, and also China. Thank you for. In this presentation, I told you about our studies on tea. I hope we will have the opportunity to work together with you on this issue. It will be an honor for us to cooperate with you. Thank you very much.
Thank you as well, respected ma'am, for your excellent and informative presentation. Now, we would like to go for question and answer. If our audience have any kinds of questions or confusion, they can ask on the chat. We will reach it to our respected speakers for the answers. So, kindly write any questions if you have.
I have two short, quick questions, if you please allow.
Sure, ma'am, go ahead.
This is Dr. 𝗖ście 𝗕𝗶𝗃𝗏𝗃. I'm looking after Department of Biochemistry and Biotechnology at the University of Faisalabad, Pakistan. Interesting presentation there. I really liked the work you presented just now. It was very informative. Two quick questions. One is when you are preparing climate resilient varieties and some genotypes there, I'm just wondering whether do you analyze, other than chlorophyll contents and other parameters, do you relate or analyze the flavor change in the tea when you have these genotypes? Do these conferring tolerance to heat, probably, as you mentioned, and some other traits, desired traits according to your climate? I'm just wondering whether altering that and introducing those resistance can alter the flavor of the tea. And the second question is, in Pakistan, we worship our tea. We begin our day with tea. So we are tea people. And I'm just wondering there, for researchers and science community, any good flavors are coming from Turkey or around where the tea is cultivated, for any good, rich in antioxidants, and same time, do we have some weight loss, some factors there? Thank you.
Ma'am 𝗕𝗂𝗵, kindly answer the question. We would like to hear from you.
Please repeat again the question. First question, please repeat again.
Yes. First question is, when you prepare your genotypes or varieties for climate resilient or heat tolerant, do you check their flavor changes? Does it alter the flavor of the tea?
Yes, yes. I tested the aroma and the other biochemical characteristic in our genotypes. Absolutely, in our breeding program, we don't focus on the stress tolerance traits such as heat or draw resistance. We also please grade and passes the tea quality, particularly flower, aroma, and biochemical composition. As you know, climate stress can significantly affect important quality-related compounds, including catechin, caffeine, amino acids such as tannin, and volatile aroma compounds. This change may alter the taste, aroma, and overall sensory quality of tea. Therefore, when we identify climate resilient genotypes, we also evaluate their quality, characteristic to biochemical analysis, sensory assessment, and in some cases, metabolomic approach. Our main objective is to develop tea variety that aren't only tolerant to climate change, but also maintain or even improve their desirable flavor and quality attributes. In other words, stress tolerance alone isn't sufficient. A successful tea variety must combine both environmental resilience and high cup quality. Thank you again for this very relevant question. Please repeat again the other question.
Thank you very much for answering my question in detail. Thank you. Second question was about tea flavors. I'm just expecting in your breeding program, should we expect some tea type coming with that is rich in antioxidant and some weight loss properties? Should we expect that?
Thank you very much for your excellent question. Yes, certainly. This is one of the important long-term goals of our tea breeding program. The natural contains a wide range of bioactive compounds, particularly catechins such as epicallocatechin, which are well known for their strong antioxidant activity and potential health benefits, including support for weight management and metabolic health. Therefore, in the future, it is entirely possible to develop new tea culture specifically enriched in antioxidant and other healthy promoting compounds. Thank you again for your thoughtful and forward-looking question.
Thank you, ma'am, and thank you another ma'am as well for doing the interacting question and answer session. I would like to request the participants as well as from our organizing committee members, if anyone has any question, kindly mention that in the chat box, or from the committee members, they can speak through their microphone as well. We will wait for a few seconds and then we will move to the next speaker.
Yeah, hi, can you hear me?
Yes, go ahead with your question, please.
Yeah, it's me, Mujahid from University of Manitoba. I don't have many much concerns with the breeding and genetics with the tea, but I have general questions. How can small scale farmers can get benefit from these genetic approaches that you are doing in this institute? And what limitations did you face in these countries?
Please repeat again, dear professor.
Yeah, I just want to know what you're doing in this institute. How can a small scale farmer can benefit by these approaches that you're performing in this institute? How can a farmer can get benefit, a small scale farmer?
Farmer is very important breeding studies because we describe different types of genotypes in different orchards. Some farmers know that which genotypes in their orchard is the best one. Therefore, we talk, firstly, we talk together with the different farmers. After that, we describe our parameters, and then we selected the best genotypes. Dear professor.
Yeah, thank you. Thank you very much.
Heartfelt appreciation to Professor Dr. 𝗔𝗴𝗍𝗕 for sharing her valuable expertise and innovative research insights. Now, it is an honor to introduce Professor Dr. 𝗖𝗵𝗿𝗀𝗍𝗕, HOD of Medical Biochemistry and Director of Research and Development at Federal University 𝗔𝗴𝗿𝗍𝗕, Nigeria. His expertise lies in medical biochemistry, natural products research. Sir, kindly proceed with your presentation. Professor Dr. 𝗖𝗵𝗿𝗀𝗍𝗕, sir.
Okay, he is facing some technical issues. Along with that, Dr. 𝗤𝗂𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 will start his presentation right now, and then we will invite Dr. Saleh Ibrahim. Please.
Okay. And now it's my pleasure to welcome Professor Dr. 𝗤𝗂𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏
Can you
Yes,
hear
sir.
me? Yes.
Yes, sir. You
Thank
can
you.
proceed with your presentation, sir.
Yes. Thank you. Thank you so much. Thank you for this organizing, such a nice organizing. Now I will share my presentation. You have no slide.
Miss Hafsa,
Yes, I think you.
Miss Hafsa, please have you shared the slide?
Yes, sir. It's presenting. Just 1 second.
Ah,
Okay.
this is not another one.
Yeah, we are sharing. Please
But yeah,
wait a
okay.
minute.
Yeah, yes. If it is
Along
possible
with
to
us.
share myself, will be better. But if you allow me, I can share with my computer. Will be very easily explanation, I think.
Yeah, you can share it with by yourself. It will be better. Here below you can see the screen
Share
sharing option.
screen. Yes. Yeah.
You can
Ah.
share the screen.
Yes. No, but how to manage this? I don't know now. Oh. I had opened in my Yeah. Okay. I will try. Wait a
It's
moment.
shared.
I will try.
So you can
Yeah, it
continue.
is. I think it is shared now.
Yeah, yeah, you can continue.
Oh, okay. I will close this one. Yeah. Uh, just full screen. You see my first slide now. The name of my presentation title is written.
Yes, sir, it's
Yes.
visible.
Yeah. Okay. Firstly, I want to introduce myself. I am professor at University of Harran in Şanlıurfa. Şanlıurfa is just border of the Syria, different city from the before presented Kezban in Rize, Black Sea region. My region is Southeast Anatolian region, just opposite of this region. And climate is also very different. For example, their climate is very rainy winter time and warm summertime, but in my region, summertime is very high temperature and no rain also. I should say firstly this one. And I am working on pistachios and fruit trees, especially pistachios, almonds, and the other fruit trees. I should change this slide, but how can do this? I don't know. Ah, you
So,
have do this. You
yes, sir, you
yes
can
you
say me to the next slide.
yeah okay. This is the meeting in Spain. It was organized 2 years before about the nut, just like a nut meeting. Not only pistachio, almond, chestnut, hazelnut, etc. Nut group meeting. There was a presenter over there mentioned that if the people produce nuts such as hazelnut, walnut, pistachio nut. This another slide, please. Uh, it is not. Huh. If they consume nut, they have a very healthy body. They will have a healthy body. It will be reduced health care, such as just cardiovascular system will be healthy if the people consumes nut groups fruits. Another one. As an introduction, I should say that pistachio is first cultivated in Southeast Anatolia, and when the Hittites people settled very before BC, 1,600 years before, and after that, it was a very valuable pistachio is accepted, and it must say that kings of fruits of kings. In fact, pistachio is not growing everywhere because of the ecological demands, I can say. Another one. But I want to, yes, Pistachia vera. I mean, pistachio is including Pistachia species. It is also growing West Asia, Afghanistan, and also in Pakistan. Some people are working there. I know them, and generally it was spreaded Caucasus, northern, western Pakistan, Afghanistan, Iran. Mainly in Iran, it is very. I should say that mother countries, and Pistachia vera is edible. The other species such as Terebinthus atlantica, Kinjuk, these are just like a wild pistachio. They can be used as a root stocks for pistachio. The next slide, please. Next slide. Yes, when we look at the general information, these are deciduous plants. This means that pistachio trees falls down their leaves in winter time. Only Pistachia lentuscus is evergreen tree, and male and female flowers born separate trees, and there is no any incompatibility between the males and females because the males of the other wild pistachios can pollinate, and fertilization may occur in pistachio, and they can be used as a root stocks. Yes, the next one. The next slide. Yes, when we look at the as a DNA composition, we can say that some of them very relative to each other. For example, Pistachia vera and Kinjo, and Palestina terebinthus, just like Mexicana lentiscus, they are very near to each other. Yes, another slide. In the world, it is not growing everywhere because of the climatical conditions. It has a very important climate conditions. In winter time, it should be cold. In summertime, it should be very high temperature. In the world, it is generally growing different countries, but in the world, the United States, Iran, Turkey, Syria, and some European Union countries such as Greece, Italy, Spain, they are producing pistachios. The next slide. The next one. Yes, in the world, the production is very important. In Turkey also. In Turkey, my city, Şanlıurfa, and neighbor city, Gaziantep, Siirt, they are producing pistachios. Another one in the world, pistachio in USA, it will be increasing because of the high yielding cultivars, and they are organizing, they are applying the very modern systems. In 2031, it will be just 900,000 tons. Yes, the next one. The next one, yes. But in 1976, the first commercial crops in California, 680 metric tons. But now, in 2023, it is just 680,000 tons. It was a very important, this spreading. Why? Because they are applying the modern system, and they are making a very good advertising. For example, Dr. 𝗏𝘓𝗏𝗏, cardiovascular expert, talking about the pistachios, and pistachio is very important for human health. Another one, please. Another slide. And it is also very advertising. Another one, antioxidants is very important. In London, on the bus, it is advertising pistachio. Why? The people consumes the pistachio nut. Yes, another one. Yeah, here, this is the Madrid and Barcelona, and some, of course, Turkey production is getting higher and higher. In the future, our production will be just follow the USA, I can say, because USA is a very modernized system. they are applying. The next one. The next slide, please. Yeah, I am mentioning about the production in the just 3 cities, 3 or 4 cities, Southeast Anatolian part of Turkey. Yes, next one. I have more slides, but the very old trees you see here, 800 years old. Another one, Atlantica. This is very important, very old pistachio trees, but mainly when we check them, the root stocks is ภิstacia king-juk. Another one. In Turkey, the white pistachio is spreading in Mediterranean, Aegean region, and Southeast Anatolian part of Turkey. Yes. Another one. There was a program or budding program for Southeast Anatolian part of Turkey. They are budded the ภิstacia king-juk to produce pistachio. Yes, another one. And the very old trees are budded by the farmers. The trainees, they were learned how to bud them and then apply it in seed, just like seed from. Another one. Yes. Yeah. This is the tree you see here. It can be grown in rocky areas. This is the characteristics of ภิstacia vera, because of their roots are very deeply goes down. Yes, another slide, please. Yeah, here you see very less soils on the surface of the rocky and calcareous soils. These are very high content of lime. Yes, mainly how to get the yield. Pollination is important. Irrigation is important. Fertilization, pruning, pest and disease management, variety, root stocks. Another one. Yes, pollination. I will mention about the pollination now. Yes, because male and female flowers on different trees. Yes, another one. But the farmers at the beginning of the Turkey in 1980, they don't believe in the male trees, because the farmer sees that male tree is not burning. It hasn't have any fruit on them. They are cutting and budding. But pistachio is known 1697. Since 1697, dioecious trees. This means that male and female flowers on separate trees. Another one. Yeah, it is necessary. The male tree should be. If it is not, the fruits will be empty. Yes, another one. The farmers are asking how many male tree for females. Mainly, we are saying that 8 female, 1 male tree, or another one, or 11 female and 1 male tree should be in the orchard. Yes, another one. And this is the male tree. Male tree should spread a very high amount of pollens. Yes, another one. If it doesn't, no. And in horticulture, we say that protandry. Protandry means that the male trees are opens the flowers before the females. Look here. This is the male tree, opens the flowers, but the females are sleeping. There is no any pollination. This should be some synchronized to each other. This is the male tree, This is the female flowers. Yes, another one. Yeah, this is the male tree. Another one. Another one, please. Yeah, yeah, this is here. Look, please look at this picture. The female trees, and there is very near. This is the male tree. There is, of course, there is no any fruit on male tree. But beginning of the in Turkey, in some Syrian people, looks that these trees are not bearing, and they are cutting this. But nowadays they have learned in the orchard, male tree should be in the orchard. Another one, please. Yeah, here just the clusters full of fruits, because there is enough male tree and pollens comes to flowers on the these female trees. Another one, please. Yeah, this shows that one flowers, pistachio flowers, this is just the length is 2 millimeters, and very large stigmas has, because pollination will be provided by wind, no bees. Yes, another, please. Another one, please. And when you look at the beginning of the fruits, the zygote is here, and the pericarp is growing. Firstly, when in middle of the June and end of the July, it will be just filled. Yes, the irrigation system, we are just advising irrigation system when the zygote will be start to grow. And if you have not any enough male tree, you will make artificial pollination. Yes, because changing of the climate changing system, the male trees opening flowers not stable. Some of the trees are, this year is okay, but another year is opens the flowers later on. Yes, another one, please. The other slides. You see here the pollens of pistachios. Yes, another one. Yes, pollination is necessary to fruit set, sufficient fruit set. Yes, another one. You see some of them artificial pollination during applying. The farmers are artificial pollination. Another slide, please. And pollens should be alive. And pollens of the pistachio to keep them in a refrigerator or deep freezer very short time. For example, in this here, the red one are alive, and the another one is not alive. Yes. Some of the farmers doing just like apply just like this. They are cutting the branches of the male, putting in a water. When the pollen shedding is started, it goes to pollinate the flowers. Yes. Another slide, please. Yeah, this method. Another slide. And some of them apply just like this. Another slide. They are putting in a floor. Yes. In Greece, this slide from Greece, they are just collecting the pollens and spraying on the trees. just like a vacuum cleaner system. Yes. Another slide, please. Yeah, here. The ventilator, they are using ventilator behind all the male trees to spread the pollens in the orchard. Yeah, another slide, please. Yeah, but pollination by wind, of course, okay. And we can apply artificial pollination, but main things is suitable male and female cultivars. We have determined some male trees for our female trees. Another slide, please. Flowering time should be synchronized. Irrigation, another problem is irrigation. Why USA production is very high? They are irrigating. Yes. Another slide, please. Irrigation increases 35% of yield. Another slide. And irrigation is very important. It is also prevents the alternate bearing. Pistachio alternate beared fruit trees because. And splitting right, fruit size will be increased also. Another slide, please. Irrigation, when I have mentioned that the zygote will be divided and will be start the growth. This is the just June. In June, there is no rain. Pistachio producer part of the these countries, and irrigation should start. But which irrigation system? The drip irrigation system is very important. Photosynthesis rate will be increasing 32%, and but very important things is water should not be touch the water trunk, because otherwise the phytophthora disease will occur. Another slide, please. Yes, in Turkey and in Syria there was no irrigation, but mainly just last 7 years in Turkey it has been started to irrigate. 10% of the pistachios are in irrigating because high incomes, because of the high incomes. But in Iran, in USA, irrigation is using. Next slide, please. And when we start, this is the pericarp growth, and at the beginning, and zygote is just here. It will be started. This time, if you irrigate in this stage of the growth, the embryo development will be rapidly, and the fruit will be splitted. The next slide, please. Here, same one. Next slide. Yeah, I have shown before in this part, it has been started. Yes, the embryo, shalaza, yes, and drip irrigation. We advise drip irrigation because of the very important point. The water should be used very efficiently. Yes, next slide, please. Yeah, next slide. Yeah, drip irrigation method are using in Turkey now, or underground drip irrigation. The next slide, please. The next slide. And there is an advantages of underground drip irrigation system. Next slide. And kernel development will be start if you irrigate very well. Next slide. flow irrigation can be done in Iran, just this method, but we are not advise this. Only drip irrigation we are just advising. Next slide, please. If you want to get such a good clusters, you should irrigate them. Yes. Next slide, please. Yes. Another one. Next slide. If you irrigate more, the plant will be dry. There is here some diseases start. Next slide. Yeah, ecology is important, especially chilling requirements, spring late frost, summer temperatures. These are very important. Our research are made on this subject. Next slide, please. Next slide, please. Yes, climate change in cultivation. For example, in winter time, if the weather is very warm, insufficient chilling occur. Pollination and fertilization problems will occur. High temperature stress in summertime and drought stress, increasing damages of disease and insect problem will increase for climate change. Yes, next one, please. Climate requirements, very important. Low temperatures, very important. And chilling requirements, very important for the pistachio trees. Next slide, I will mention about some, yes, chilling requirements. The bloom is not enough if it's a low temperature. Yes. Another one, please. We have calculate our cultivars and, for example, our cultivar chilling requirements. This is very important for climate change. If the winter will be warm, for example, ohadi will not grow. If the inadequate chilling problems will occur, yes. Another slide, please. Yeah, drip irrigation, we are just advised. And if the insufficient chilling, the leaves are not enough. Next slide, please. Summer temperature should be very high and sufficient because the splitting and hard shell they hold easily. Yes. Another slides. Yes, water irrigation, winter irrigation. This means sometimes in winter there is no rain, and hailstorm is very important in springtime. You see here. Next slide, please. And the flowers will be drops here because some hailstorm and flooding may occur. Next one, please. Next one, please. Next slide. Yes. If the irrigated ones, drip irrigation. Next slides. In California, just like this irrigation system is applying. In Greece, just like this irrigation system. The next slide, please. In Greece, underground drip irrigation system, water save for this system, and fertilization. A little bit fast, please. Another slide. Another slide, please. Fertilization increases 36% of yield. Another slide, please. And soil and leaf analysis is very important for pistachio, to make a program for fertilization, and the leaves should be taken analysis in July. Yes, another slide, please. And some of the fertilizers should be given around of the tree, just like this. Another slide, please. yes, application of the yes, on rocky areas, it can be grown. You see here some roots. The roots are system is very important. Pruning, yes. Another slide, please. The orchards are getting higher and higher. Hand pruning. In Turkey, we are applying hand pruning. Yes, another slide. In USA, mechanical pruning. In USA, yes, mechanical pruning is they are applying. Mechanical pruning, yes. Another slide, other slides. In also mechanical pruning, using in Spain in almonds. These are almond trees. Yes. Another slide, please. Yes, they are preparing the trees for mechanical pruning because of the labor is very important. Yes, another factor is cultivar, different cultivars in yes, different cultivars we use in Turkey. We have registered the new variety. Yes, another slide, please. And Tekin variety is very famous in Turkey also. Yes. Another slide. Sear, Tekin, 𝗔𝗴𝗿𝗃𝗃𝗵. These are our cultivars. And we have worked on them, different cultivars and yield. Yes. Another slide, please. And root stocks is another important. Generally, we use 𝗵𝘗𝗃 𝗕𝗔𝗃. We recommend 𝗵𝘗𝗃 𝗕𝗔𝗃 is very important. Secondly, 𝗵𝗲𝗃 is important. Yes. Another slide, please. Root stocks, yes. These three, very old tree, 𝗵𝘗𝗃 𝗕𝗔𝗃 and root stocks, 𝗵𝘗𝗃 𝗕𝗔𝗃. We recommend very well root stocks for pistachio. As a root stocks, UCB1 from USA, we are also using this one also. Yes, another other slide, please. In the mountains, they are also growing 𝗵𝘗𝗃 terebinthus. These are 𝗵𝘗𝗃 terebinthus, very hard conditions. They are growing and very good orchards. And they are also using the vineyard, the new orchard. Other slide. Calcareous soil, very bad soil. We are budding them. Yes, another slide, please. And we are growing in the pot and budding. Other slide, please. And newly established orchard now. Other slide, please. And we are also budding them. Budding is very important. And the 𝗕𝗔𝗃 𝗵𝗲𝗃 is very high yielding root stocks when we make a comparison. Yes, we are training the farmers also. Yes. Pest and disease are very important. Yes, the 𝗵𝘗𝗃 and another one. Other slides. The same one is coming now, but should be septoria. Yes. Yeah, we should consider this one. Yes. Other slide, please. Very important. Other slide, please. Other slide, please. My voice is coming or no? Other
yes,
slide.
you are audible.
Yeah, okay. Other slide. Yes, same one is turning back. Yeah, okay. I think my time is finishing. Phytoplasma problem. Yes. Please go fast. Woodpecker. Woodpecker is very important bird. Yes. And harvesting. It is finishing now. Harvesting by hand in Turkey, in Iran, but in USA. Other slide, please. Other slide, please. In USA, by Mason. They are doing this by Mason. Yes, storing is another important. Yes. Other slide. Aflatoxin problem in the world, it can be occur. Other slide. Yes, our famous seed cultivar, and we should consider this, and we should store in a very good system. The result irrigation is important and processing is important. Good agricultural practice is very important for fruits. Yes, other slide, please. In Turkey, there is a 200,000 family is just consuming using the pistachio. Yes, thank you very much for your attention, please.
Yeah, thank you,
okay.
sir, for
Thank
your
you.
deep.
Yeah, other slide. Could you bring other slide, please? I think another one, the last one. This is my city. This is very ancient city. Hazreti Abraham was born in Şanlıurfa, and this is the Atatürk Dam, and we have a 𝗂𝗏𝗃𝗃. 𝗂𝗏𝗃𝗃 is 12,000 years old civilization in Şanlıurfa. This is my city. Thank you very much for listening me. Sorry, I have extended a little bit. If I check my slides myself, it would be just on time. Murtaza. Thank you, Murtaza. If there is
Thank
a
you, sir.
question, yes.
Thank you so much.
Thank you so much for your presentation. Due to our time shortage, we would like to move to the next speaker, which is Professor Dr. 𝗂𝗏𝗿𝗀𝗿, HOD of Tropical Fruit Cultivation Technology at the Scientific Research Institute of Fruit and Tree Growing, Azerbaijan. His presentation topic is pathogenic microorganisms that cause reduced yield and deterioration of quality in plants. Sir, kindly take the floor. Professor Dr. 𝗂𝗏𝗿𝗀𝗿, are you there? Sir, Professor Dr. 𝗂𝗏𝗿𝗀𝗿.
Yes, I am here. Do you hear me? I am here.
Yeah, you are audible. You can open your camera and you can share your slide is visible, and you can continue. Yeah.
Yes.
Okay. Please set your mic. Your voice is little bit distorting.
Do you hear me?
The microphone is so low, ma'am. Can you?
Okay, do you hear me? I am here.
Yes. Yes.
Okay. Okay, I will continue. Okay, if no problem.
Miss
Um,
Ǹokpotr, please please
Dear
invite her.
Congress participants, since the time allotted to the participants is 20 minutes, I will try to speak within the given times. My main research area is study and detect microorganisms that cause pathologies in plants, clinic measure to combat, and so on. Next presentation, please, slides.
Ma'am, a little confusion, which is your voice is so low. Can you come closer to the mic so that we can hear you properly? And another thing is, please keep the time reminder so that you can finish on timely. Thank you.
I am sorry, I can't hear you.
Ma'am, kindly come closer to the microphone so that your voice is more louder. That was the question.
That's right.
Yeah. Is it clear? Now clear,
Yes.
please. You
Okay.
continue.
Um, then I will continue. Okay. Issues such as constant increase the number of populations within a fixed area, for example, wars, natural disaster, continuous human interference within nature, and so on, among the factors that cause serious food shortage. This is one of the serious problems that must be solved in the current area. Next, next. Thank you. According to an assessment conducted on a climate change model, model, it is noted that a climate change could cause. Next slide, please. Next presentations, please. Okay, thank you. Climate change could cause 15% yield loss in corn productions in the United States by 2050, which means a financial loss of 6.7 billion. In addition, changes are also occurring in the fungal biotope that inhabits soil, water, and plants against the background of climate change. If we take into account that many microorganisms plays an indispensable role in soil sanitations, antagonistics to pathogens and plant symbiosis, then there is no need to say how serious this issue is. Such situations result in the spoilage of tons of plants based products used by humans as food due to problems caused by microorganisms that can lead to food shortage for humans. I can't see presentations. Can you turn it?
Kindly, the moderator, share the screen again.
Um,
Yeah, please, 1 minute.
do you hear
Just
me?
please. Yeah, please 1 minute.
Um, do you hear me?
Yeah, you
Uh,
are audible.
yes.
Yeah, please 1 minute. Is Nurpur, am I audible?
Yes, you were audible. Yeah, kindly share the screen.
Can you show my presentations, please?
Yeah, I am sharing. Just wait a minute.
Not this one.
I can't see my presentations. Can you show it me?
Yeah, I am sharing. Just wait a minute, please. Yeah. Am I audible, Nurpur?
Yes, you are audible. Kindly share as soon as possible. That would
Yeah,
be
we are
better.
I, I am sharing. Just keep in touch with our guest speaker, respected guest
Okay.
speakers.
It is really nice to meet you all, and we were having a small
My
technical issue.
presentations, please.
Ma'am, we were having a small technical issue. That is why your presentation were cut off in the middle of the presenting. We would request a 1 minute or maximum 30 seconds if that's possible so that our CEO can share the screen. In the meantime, we can have an interaction such as the presentation topic, about the presentation topic we were about to discuss, as you were about to present as well. So ma'am, can you describe in the meantime? We would like to hear some insights, such as it is the presentation based on the pathogenic microorganisms that cause reduced yield and deterioration of quality in plants. So I think this presentation will provide important insights into plant disease, microbial threats, and their impact on agricultural productivity and food quality. So what is the difference of having diseased plant and having healthy plants? Can you give us a little bit insights of the differences between them?
Nurfu, I am sharing the screen. Please check.
Yes, kindly.
Is screen visible now?
Not yet. It's loading. Yes, it is visible now.
Yeah, you can continue.
Ma'am, the floor is yours. The presentation slide is also showing. Kindly proceed with the presentation. Professor Dr. 𝗂𝗏𝗿𝗀𝗃, ma'am.
Antonia, do you hear me?
Yes, you are audible.
Okay.
Kindly proceed with the presentation.
Four presentations, please. Four.
Slide
Okay.
number 4, please.
Yes, 4 slides. Yes, number 4. Okay. In
Okay.
addition, changes are also occurring in the fungal biota that inhabits soil, water, plants against the background of climate change. If we take into account that many microorganisms play an indispensable role in soil sanitations, antagonistic to phytopathogens and plant symbiosis, then there is no need to say how serious this is. Such situations result in the spoilage of tons of plant-based products by human as food due to problems caused by microorganisms. This can lead to food shortage for humans. Next. Okay, thank you. At the present time, the increase in the number of people and their purchasing power has also attracted the interest of people working in the agricultural and scientific research fields. Experts working in this field are carried out a number of works to ensure that the products is both quantitatively and qualitatively high. First, taking measures to combat diseases found in fruits and berry plants, providing correct diagnostics of disease pathogens, microclonal propagations, and breeding disease-resistant varieties. Also establishing super intensive gardens using modern technologies. One of the main goals of our research in the timely detect of microbial pathologies that cause a decrease in quality and productivity in plants, and the timely implementations of preventive measures against them. Next, please. Okay. Among the mentioned issues, this is found in plants and their timely detections and proper control measures are very important in terms of both quality and product times and the preventions of crop loss. There are diseases such as powdery mildew, anthracnose, septoria, wilt, spotting rash, alternosis, and other diseases which cause myths in the plants. To date, based on samples, next please, taken from various fruits and vegetables, we have recorded mainly the disease and their pathologies listed in the table. You looks how you watch from the tables. There is disease, their pathogens, mainly fungi, which is located in that plants and cause different diseases and disease prevalence. Next, please. And there is next, please. And there is also the persons of the disease which found in the fruit plants and berries. Next. Okay, thank you. The table above shows the distributions. There is shown the number of fungi recorded in the subtropic fruits, which we take samples from them, and we learn microbiota of the fungi, bacteria, and fungi which located in that plants and which they cause different diseases, how we said about it. And there is, next please, the table above shows the distributions of microorganisms recorded in subtropic plants according to substrates specificity. There is given universal relative substrate specificity which have and relatively specific substrate specificity fungi. Universals are those that are found everywhere, all fruits and plants. With relatively substrate specificity, there is fungi which, as the name suggests, it is relative, and those with substrate specificity are those that lives only in the place where their food, there is the plants. Next, please. Next, okay. Apple, cherry, and hazelnut are some of the most widely cultivated and economically profitable plants in Azerbaijan. Phytopathogens recorded in these plants not only reduce their quality and quantity, but also cause serious economical problems if errors are detected. Next. The most widespread and seriously damaged fungal disease of plants are black cancer, powdery mildew, spotting, and fruit rot. The pathogens of the listed fungal disease develop mainly, next please, mainly in the spring and summer months, especially unstable visa, rarely visa, and suitable visas for the microorganisms, and they are given in the slide. Slide, please. Slide. Next slide. No, next and next. I said about them. Next, okay. Others also, next. Thank you. In this slide, shown the most common disease in cherries and their pathologies, which was given in that slide. Next, and in the next slide, there is was shown the most next, please. Yes, no, yes, that's the slides was showing the most common pathologies in hazelnut. And next, next, other slide. Okay, they are with taking samples from the hazelnuts. And the next slides. Next, please. Other slides. Okay, some of the pathologies recorded in plant are produce of mycotoxins, which is very dangerous for the human health. One of the plant is hazelnuts, which we said, and widely used by human for various purposes and providing economic benefits. If microorganisms that produce toxins are detected in these plants or its products, this can lead to serious problems. These microorganisms, including species of that is Aspergillus flavus, Aspergillus parasiticus, and Aspergillus niticus, belonging to the genus of Aspergillus. They are produce aflatoxins, which I said makes serious problems for human health. Next. Other slides, please. Okay. Okay, thank you. How you seen in that tables, there is given mycotoxins, fungi which produce mycotoxins. For example, we said about fungi from the genus of Aspergillus. There is given Aspergillus flavus, Aspergillus parasiticus, Aspergillus ochraceus, which they produce. For example, Aspergillus flavus and Aspergillus parasiticus, which we meet in the hazelnuts, peanuts, and corn. They are producer of the aflatoxin B1, 2, J1, and 2, and also ochratoxins A, and they are most dangerous toxins which produced by fungi, mold fungi. Next, there is the next, please. Other slide, please. Okay. No. No. Okay. No problems. Yes, that's problems. Yes, that's. They are given fungi which we take from the plants and cultivate in the nutrient media in the petri dishes, and the morphological images and other images. There is fungi Aspergillus under microscope. Next, and we said about next presentations, and we said about aflatoxins, why they are so dangerous. Next, please. Presentations. Next. Also, yes. There is was shown where they may spread, and that plants, crops, we mainly use everywhere in our homes, and food feed for animals, mainly cow, and we meet that problems. We may meet that problems everywhere, and the effects in the human health.
children developments, immune systems, and everywhere. Next, and there we said more dangerous. Aflatoxin is an organic compound with lethal effects. Next, please. Next slides. It is a biological contaminant. The most dangerous forms is aflatoxins. There is the B1 representative of the genus Aspergillus, which produce aflatoxin form, then white thread like cluster, which later change color due to the maturations of spores. Since aflatoxins is highly toxic, its content is strictly controlled, and the permissible limits for its content in plants are indicated in the table. Okay, in the table, thank you. Maximum limits of aflatoxin is hazelnuts products. There is shown how you say hazelnut intended for direct human consumptions for or as an ingredients in food products, only for the aflatoxin B1. For 1 kilogram, it must take no more than 5 milligrams, but general amounts of the B1, B2, J1, and 2, 10 milligrams in a kilograms, and no more. And about all the forms, it's shown in the tables. Next, please. In this regard, we conduct regular monitoring to ensure that the quality of the products is higher, and that any problems that may arise are detected in a timely manner. These monitorings are important in preventing the expansions of the problems that may arise, and in the storage of the products after harvest. For the determinations of aflatoxin in fruits after harvest, two main solutions have been developed: immunochromatographic analysis device and enzyme like immunosorbent analysis ELISA. Next. Next, please. Other slide, please. Okay, thank you. Taking all this into account, aflatoxin analysis is being carried out on hazelnut plants at the Molecular Biology Laboratory of the Scientific Research Institute of Fruit Growing and Tree Growing of the Ministry of Agriculture. And I want to say that we are continuing our monitoring to taking samples and to made how to made measures and have biopreparates to the, take control that problems. Thank you for attentions.
Thank you so much, Miss, for your deep and insightful presentation filled with informations and insights. Now we will move into the question and session. If anyone has any question, kindly mention in the chat box. Okay, I think no one has any question. So thank you very much, ma'am, for your insightful session. And it was a nice presentation filled with lots of insights and information, helped us to know about the yield and the storation of plants. So we will like to invite our next presenter.
Yes, thank you very much.
You are welcome, ma'am. So now it is an honor to introduce Professor Dr. Salihu
Audible?
Ibrahim Ismail. Yes, you are audible.
Okay. Okay. Thank you
May
so
I
much.
proceed?
Thank you so much, Dr. Kenal Bhksholai, I really appreciate your work in the domain of plant protection. Nice to meet.
Can I go ahead?
Yes, 𝗏𝗏𝗏𝗏𝗏, please invite him. Yeah.
Yes, yes. It is now an honor to introduce Professor Dr. 𝗤𝗵𝗿𝗄𝗏, HOD of Medical Biochemistry and Director of Research and Development at Federal University 𝗔𝗴𝗿𝗄𝗏, Nigeria. His expertise lies in medical biochemistry, natural products research, and biochemistry innovation. Today's presentation topic is the evolving role of natural products in the 21st century. Sir, kindly proceed with the presentation. The floor is yours.
Okay. Good day, ladies and gentlemen. I am presenting on the topic that has just been introduced, the evolving role of natural products in 21st century drug discovery. Next slide. I don't know, can I control my slide or I request? Very good. So as you can see from my first slide, all the natural compounds, the organic molecules that we use in drugs, initially they come from plants, because right from the onset of time, plants were the major source of drugs used by humans in ancient civilization. But with the development of the knowledge of chemistry, advancement in research techniques, and the use of tools make it possible to extract, purify, isolate, characterize, and structurally elucidate these organic compounds that we make into active pharmaceutical ingredients that we incorporate into our present drugs. So in a nutshell, what I'm trying to say is that all the organic compounds that we use initially, they come from plants. And coming from plants, we use the synthetic template of some of these organic compounds, because when we extract and isolate them, the synthetic template now will serve as a backbone where we build on the conventional medications. Next slide. So as you can see, from the willow bark, the drugs that initially we used to treat pain, aspirin, the poppy seeds that mostly used for sleep. So you can see that from my introduction that mostly the blockbuster medications that we have almost come from plants, like metformin that comes from the willow tree, digoxin, and many other ones. Next slide. So now we are in an era whereby we are returning back to nature. Of course, initially, the world has developed in such a way that we synthesized these chemical synthetic drugs. But now we have no option but to go back to nature to try to understand what the nature has offered. Because if you look into this thing, uncharted chemical space, because natural drugs occupy highly diverse geometric shapes. Because you cannot compare a drug that you isolate from plants with the one that you synthesize in the lab. Because the one that comes from plant, they are highly diverse because their structural, their carbon chiral centers are different from the ones you synthesize in the lab. And structural complexity. Most of these drugs that we extract and isolate from plants are structurally complex, structurally diverse, structurally rich in chiral centers and oxygen atoms that will provide enabling ground for binding into the receptors in humans. When you look into the third act of the evolutionary validation, over millennia, the plants have been pre-optimized by nature to bind automatically and easily without any inhibition to the biological targets. But when we use the synthetic template now, we have to always be trying, how do we bind it? How do we stabilize it? by creating the ion-ion bonding interaction, by trying to see how do we create hydrophobic interaction, how do we create salt bridges. But when you use the drugs extracted, isolated from plants, they have been pre-optimized. It means over millennia, nature has worked on them, has already developed so that they can easily bind to the human target. And of course, they are naturally capable of crossing cell membranes. Because for a drug to bind and work, it has to have that what we call drug likeness. Because it's not any chemical compound that can bind to the body and create and change the body physiology. So that compound has to have what we call drug likeness, that it has the potential at least to affect positive changes in the blood and the body. So the natural compound are naturally have this natural inherent drug likeness and are capable of easily binding to the receptors within the cellular membrane or crossing the cell membrane, as in the case of steroid drugs. Next slide. Next slide. So if you can see from this my slide, on the left side is a simple synthetic compound. On the right side is a complex natural product. Look at the richness. The red, it's indicating oxygen. Look at the complexity. Look at the richness. Look at the diverse geometric design. So you cannot compare product isolated from nature compared to one that we synthesize in the lab. Next slide. So when you look at what we are witnessing from the slide is a structural architecture. So trying to explain the previous slide, synthetic chemical libraries tend to be flat, rigid, and structurally similar. But when you go to natural products, they have a unique chemical space. They have varied chiral centers. They have varied three-dimensional complexity, three-dimensional configuration, whereby the oxygen atoms, the nitrogen atoms, the phosphorus atoms are rearranged in a way that makes them easy to bind to the receptor within the human body. And they are evolutionary pre-validated, because what we are trying to say here is like for thousands and millions of years, the plants have developed a mechanism to rearrange the structure of this compound to make it easy targetable to a receptor. And just like nature has spent millions of years perfecting these molecules to interact with the biological system. So that's why they are a better alternative to the synthetic chemical drugs. Of course, plant drugs isolated, they serve as a template, because most of the drugs that we are using now, we can look into nature, we do biomimicry, whereby we study how does nature rearrange these carbon compounds, rearrange these benzene rings, so that we design a template based on the background of nature. Next slide. So another advantage we have, the 21st century technological enablers that reshapes drug discovery. Unlike before, we rely only on extracting, purifying, we isolate. When we isolate, we start going and testing the drug, whether we get a hit or not. Like we are doing blind search. But with these technological enablers, we have genomics at our disposal, whereby we can use it to unlock the biosynthetic gene clusters of different conditions, of different diseases, of different metabolic pathways, so that we can have a targeted drug delivery system. We have metabolomics, whereby we can study the mass spectrophotometry. We can map organic compounds in real time. We have the synthetic biology, engineered even host organism, so that to express exactly what we need. And of course, with the coming of AI and machine learning, I think predicting drug target becomes more easier, more simpler, and more advanced and more target specific. So we can predict a drug target with surgical precision using this genomics, metabolomics, AI, and machine learning at our own disposal, unlike before, whereby we are doing a blind search. Next slide. So when you look into the natural product discovery pipeline, of course, at least genomics can help us sequence the whole microbial genomes, identify cryptic gene sequences, activate BGZ using specialized promoters. That one is very easy in most of the laboratories that can be done. Of course, the metabolomics too. You can have rapid mass spectrophotometry and mass spectrophotometric molecular networking. You can analyze molecular fingerprints in mixers so that you can save your time. And in static biology, that one, engineered host organism Fourier compound expression, that one is a much easier thing to do in most laboratories now. And AI machine learning, of course, like I have said, you can predict a drug target with much surgical precision. Next slide. Next slide. So, today genomics reveals that microprocesses silent by static gene clusters, blueprints, molecular targets that would have never been made in a standard laboratory. So just like I have made, still trying to explain the points I had made earlier. We can cut these genes out, pass them into host organisms to manufacture the compounds of our own interest. Furthermore, modern mass spectrophotometric metabolomics allow us to map every molecule in an extract simultaneously in real time. While using algorithms, we can predict exactly how these complex structures will bind to human disease targets. Next slide. So, the modern isolation and screening methods. We have a concept called dereplication, whereby we instantly identify and filter out known compounds. You know, in traditional drug discovery, what we do, we end up extracting, because in a particular plant specimen, you can extract as many compounds as you can. From 10, 20, hundreds of plants, even thousands. So it's when you come to the purification, the isolation, that's when you are trying to look which compound of interest you are looking for. But in this, instead to be replicating, getting the same compound that you can characterize, you can instantly filter out non-compounds or compounds that have structural similarity to compounds that already are known, so that you can go directly with compounds that are unknown, like virgin compounds. And virtual docking, of course, you can simulate millions of molecular interactions computationally without having to start testing the in vitro drugs and other things. And microfluidics, droplet-based high-speed extract testing, or like the one that we keep using, columns packed with silica gel and everything. And this new cryo-EM imaging, the atomic level drug target contact points. So like you can even see the drug to the receptor, like SAR, structure activity relationship. So with this imaging, you can establish and visualize the contact points at which your drug candidate establishes relation with the receptor. Next slide. So I just try to give a brief holographic drug discovery pipeline, whereby, of course, number one, you have to identify the target, which is target identification. You identify the protein receptors that are interested. Before you identify the protein receptors, you have to even know which disease are you targeting. For example, let's say like you are targeting hypertension, high blood pressure. So what are the target proteins? Angiotensin converting enzymes. So what do you want to do? Do you want to activate or inhibit them? Or I want to inhibit angiotensin converting enzymes. Okay, then you study. Then you have identified, okay, this is the angiotensin converting enzyme, the protein, that is the target identification. Then you come to the drug screening. You begin the screening, the natural compound library, whereby you can find which compound could give you a very good SAR, structure activity relationship between the drug candidate and the receptor. And once you move to the next step, whereby you need the lead optimization. Is there anything that I can do to this target? Maybe addition of methyl group, addition of nitrogen compound, or removal of oxygen. What do I do to optimize the drug candidate so that I can optimize it? It can have a very good binding and a stability and affinity, because it's not just enough for a drug to bind to the receptor. It must be stable. It's just like you riding a bicycle. Of course, somebody can ride a bicycle, but if you cannot stabilize and float, you'll not be able to enjoy the riding. So, like, stability is one of the most important things when it comes to drug lead optimization. And of course, molecular validation, if you can see from the number 4, the human receptor protein and the natural product drug candidate, they are binding on the surface of the lipid bilayer of the cell membrane. Next slide. So, in a nutshell, the old way of finding drugs was very slow. You pick a plant, extract, separate, test, only to find out you discovered a caffeine. But of course, just like I mentioned earlier, now you can use the replication process where you can filter out non-compounds and go directly for non-compounds. Using liquid chromatography coupled with mass spectrophotometry databases, you can instantly ignore non-compounds, focus only on novel chemistry, virgin compounds that have potential to give you what you are interested in. So we can combine virtual screening, where millions of digitized natural structures are computationally docked into disease protein so that you save more of your time. Next slide. So, of course, we have so many druggable targets. In oncology, in cancer, marine-derived toxic payloads for targeted therapies, antimicrobial resistance, neurodegenerative diseases, immunomodulation. These are all areas that require new drugs, new drug candidates that come from natural products. Next slide. So, no. So we just take a case study. Okay. You are set. Okay. Now, what I'm trying to show here is like we can have a targeted. No, go back. Previous slide. Previous slide. Next. So, next slide. Yeah. So, what I'm trying to show here is like you can see these are the cancer cells, like what we said that you can in oncology, we have a lot of opportunities there. So this one is a targeted gluing capsules. For example, drug delivery vehicle nanocapsule emitting targeting signals and breaking apart cell integrity. So you can visualize mostly tumor mass, the cellular structures like real time when you send these natural products or drug-derived candidates from plants into this. Next slide. So briefly, we have like marine organism from the deep ocean, which is an example I gave. But the last complex plant polyphenols are also found to possess unique multi-target mechanism required to tackle neurodegenerative diseases like Alzheimer's, where single target centered drugs have repeatedly failed. Because what the point I'm trying to make here is like plant-derived natural products. They tend to have, they don't have like a single target. They can have multiple targets, and they can cause a change in body physiology, not through only one mechanism, but through diverse and varied mechanisms. Next slide. So like case study, Taxol. Of course, we know it was originally Taxol paclitaxel is an anti-cancer medication, which originally was isolated from the bark of a Pacific yew tree. Of course, mechanism, it freezes the cellular skeleton, stop cancer cell division. That is major effect. But every production requires you to destroy thousands of rare trees. So before you produce taxol from organic compounds from nature, you can cut not thousands, but sometimes even tons of trees to get an appreciable amount of this taxol. But of course, when we understand the structure of taxol, we can synthesize in the lab instead to be cutting trees, just go cutting trees, and those trees are very rare, and those trees are not even found, are not even common. So imagine you keep cutting trees just to get some few grams of taxol, which you are shifting the balance of ecosystem. But with first century technologies, we can extract, purify, isolate, characterize, elucidate the structure of that compound. And we can use sustainable bioreactors to produce that same taxol close to the natural environment it's found in plants. Next slide. So you can see, I give an example of the plants by the left. So these bioreactors are very much easy, whereby when you understand the structure of the compound, you can use bioreactors and synthesize with a close similarity to its own natural environment in plants. Next slide. So this is just brief introduction about the taxol, which I have said can produce cleanly and sustainably using plant cell fermentation in large bioreactors, proving how technology bridges gap between nature and commercial medicine. Next slide. Case study artemisinin, which is sourced from sweet wormwood based on ancient herbal text, and frontline global treatment for malaria, which is very, very known to be a component of antimalarials. But agricultural yield changes based on climate and geography. So sometimes the concentration of artemisinin in plants tend to change due to climate change, environment, soil type. But with the 21st century technologies, engineering the metabolic pathways into 𝗏𝗃, so it's easy for you. You can use technology to what? To engineer how you can produce this same artemisinin in these cells, so they can produce this artemisinin for you. Next slide. So sample photo whereby you can produce this artemisinin from these cells. So you can see the body in cells, the artemisinin molecule in green that has been produced, and you can extract it, you can purify and crystallize it. Next slide. So now, what are the challenges? So we have two things: industry obstacles and future solution, whereby we want to see you have to balance the two. Of course, industry obstacles, sometimes we have strict regulatory compliance, manual reporting errors, resource and biodiversity limits, climate change, complex global agreements. All this affect how you use natural products, because some countries tend to have stricter regulation when it comes to the use of plants. In fact, some countries even prevent cutting trees. They have different regulations. And some countries, you have the trees, but because of climate change, harsh weather conditions, soil types, acidity, will not allow those plants to concentrate that active API of interest. But the future solution is easy. With the use of technology, you can use predictive compliance systems, automated and accurate data analysis to try to understand your own isolated purified compounds. You can use data-driven impact modeling, AI-powered monitoring, and smart. Sorry, this city shouldn't be there. But you can use AI-powered monitoring, whereby you can develop models that mimic nature. Next slide. So The future of pharmacology is genome-driven intelligent discovery, so nature is no longer viewed as a static repository of plants, but as an expansive digital database of genetic blueprints waiting to be unlocked by modern technology. Thank you.
Thank you so much, sir, for your influential, informative, as well as a deep, insightful session. We are really pleased to have you as our speaker. Now, we would like to go for question and answer. If anyone has a question, kindly write in the chat box. As we have time shortage, we will not move forward fast. Okay, I guess no one has any questions. So now we would like to welcome Dr. 𝗦𝗂𝗀𝗶𝗃𝗏𝗏, Associate Professor and Scientific Research Leader from the Scientific Research Institute of Fruit and Tea Growing, Azerbaijan. His presentation topic is Genomic Diversity Assessment of Fruit Plant Genetic Resources in Azerbaijan. Sir, if you are present, kindly take the floor.
Thank you very much. Thank you very much. I will try to control my presentation if I am available. Now, I want to screen my share.
Yes, sir, it's sharing. Just 1 second.
No, here you share my presentation.
Sir, do you want to share it yourself?
I tried, but I can't.
Okay, our moderator will share it from your side. Don't worry.
I am sharing.
Okay. Okay. Do you hear me?
Yes, you are audible and the slide is also showing. Kindly proceed with the presentation.
Yes, thank you very much. Dear colleagues, dear participants, dear friends, first I want to thank the organizers for this valuable conference. And I do believe this conference will open new doors for collaboration. And I hope in the future we will organize this conference in Azerbaijan or Pakistan or Turkey without any technical problem, and we will know each other well, and we will do close collaboration in the future. Next slide, please. As you know, Azerbaijan, our region, including Turkey, Iran, and other countries, this area is the gene center. It's the gene center of a lot of crops. And Azerbaijan has great biodiversity because we have very diverse climatic condition. And as you know, in Azerbaijan, we have almost 9 climatic zones. Therefore, there are a lot of plant species. In Azerbaijan, there are almost 5,000 higher plant species. And this amount of species is equals 65% of plant diversity of Caucasus region and 11% of the world's flora. And next slide, please. In Azerbaijan, we have also a lot of crop wild relatives. And our scientists have identified that there are almost 1,200 plant species we call crop wild relatives, which we can use in agriculture. And we have done a lot of expeditions on the cereal plants, legume plants, as well as medicinal plants. And Azerbaijan is the motherland. Not only in Azerbaijan, our region is the motherland of these crops. And next slide, please. If we talk about the fruit plants, we have also very rich diversity of the fruit plants. And in Azerbaijan, we have almost 150 fruit crop species, and a lot of wild fruit plants also. I want to say in Azerbaijan we have wild pomegranate, wild figs, apple, and etc. Therefore, we also say Azerbaijan is not only motherland of the cereal and legume plants, also it is the origin country of a lot of fruit plants. Next slide, please. And we have done a lot of expeditions along Azerbaijan, and 2-3 years ago, within one project, we have identified 6 genetic hotspots. And you can see on the map of Azerbaijan, we have identified, and this region, you can find a lot of higher plant species, including fruit plants. As you know, a few years ago, we have liberated territory in Karabakh. We couldn't organize the expedition to Karabakh region. Inshallah, in the future, we are planning to do expedition to Karabakh also. But from reference, we know also there are a lot of hotspots in Karabakh region, and hopefully in the future, we will increase the genetic hotspots of Azerbaijan. I can say we will have at least 10 hotspots. But when we have the expeditions, we observed that every year we are losing some plant species because of the climate change. As you know, climate change is the huge problem. 2 years ago, we host COP29 in Azerbaijan, and we had a lot of guests, a lot of scientists. We discussed climate change problem, and they mentioned that temperature will increase 1.5 Celsius degree until 2050. But here I want to inform you, in Azerbaijan and also in our region, temperature already increased 2.2 Celsius degree, and end of this century, temperature will increase almost 4.2 Celsius degree. And therefore, we are losing a lot of the species. We think conservation, maintenance of all the biodiversity, including crop wild relatives, are very crucial for Azerbaijan and other countries. Next slide, please. And next slide. And conservation, biodiversity conservation is very important, and we have started biodiversity conservation almost 20 years ago because, as you know, in Soviet time, we maintained all our accessions, all our genetic resources in the Russian Plant Industry Institute. But 20 years ago, we started to conserve all our materials in different stakeholders, in different institutes of Azerbaijan. Now we have conserved almost 20,000 accessions in different institutes. And if we talk about the fruit plants, we maintained 4,000 accessions in the field gene banks of the Fruit Plant Institute, and we are trying to phenotype all of them. And next slide. And it is our institute. It is located in the north part of Azerbaijan, and this institute has a lot of experimental station and field gene bank, and we are trying to conserve, to maintain all our resources in the experimental stations. Then we need to evaluate. Conservation, of course, is very important, but we need to characterize and also we need to evaluate all our genetic resources for breeding purpose, for agricultural purpose. Therefore, we are trying to use international protocols. As you know, Biodiversity International have developed a lot of descriptors for many crops and including fruit plants, and we are using the international descriptors for evaluation and characterization of our genetic resources. And next slide. And but my group, we study also genetic and genomic diversity of our resources, and we started with the pomegranate. As you know, we have very rich pomegranate biodiversity in Azerbaijan, and pomegranate is one of the symbols of Azerbaijan. And we have almost 200 land races. And in our experiments, we studied genetic diversity of 85 pomegranate varieties, and we observed very high diversity, high polymorphism. And in 2023, we published our data in the Russian Journal of the Genetics. And then we started to do a little bit deep work. We started whole genome sequencing project in Azerbaijan, and we have selected one variety for de novo sequencing, and we did also transcriptomic analysis on one variety. And next slide, we have done a lot of analysis. We did whole genome duplication analysis, gene sequence, gene set comparisons, and etc. And we did also a differential expression analysis. And we have found almost 500 gene family in the pomegranate genome, and we compare all of them with the other crops, mainly Prunus persica, Vitis vinifera, and etc. And the next slide, first time we also identified SSR types. As you know, it is the molecular markers, SSR types and SSR motifs, and we identified, and hopefully in the future, young scientists will use these types of molecular markers for identification of the pomegranate varieties. And the next slide, we did also first time we did also gene expression analysis, and we observed that genes of the roots expression level is the higher than other organs, and also we observed the same expression level in the young and a little bit the old stem. And then we started, first we did de novo sequencing, only one variety. Then in the world, first time we started pan-genome project on the pomegranate. We resequenced of the 6 additional pomegranate varieties of Azerbaijan, as well as we compare all our data with the Chinese and the Tunisian pomegranate data, which we have got from the NCBI, and we identified core genes, specific genes, and etc. And then we analyzed the genomic diversity of them, and we did also association mapping, association with some economically important traits. This is the next slide, please. And it is very deep research, and we got a lot of good, interesting scientific results. If you want to get more information about our pomegranate project, you can find our paper from the Frontier in Plant Science. 2 years ago, we published our data in Frontier in Plant Science. And the next slide, then we started another project within the PhD project. Pomegranate project also was the within the PhD program, Sabine Hacıyeva's PhD work. Then we started another PhD students research, 𝗕𝗏𝗿𝗀𝗍𝗏, and we started to study genetic diversity of the fig accessions. In Azerbaijan, we have a lot of cultivated fig varieties. We have almost 70, 80 varieties. In Azerbaijan, we have also wild fig species, and mainly distributed in the south part of Azerbaijan. There is one big park. And we studied genetic diversity of them for wild and also for cultivated, and we published 2 papers in the Springer Journal. And last year, in 2025, we did also a resequence of 50 fig accessions, and we identified almost 5,000 high-quality SNPs. And using these SNPs, next slide, please. Using these SNPs, we tried, we clustered all our accessions, and here you can see Azerbaijani figs mainly belong to West Mediterranean gene pools. And as I mentioned, we have used the wild and cultivated. And in the next slide, you will see we slightly differentiated wild accessions and also cultivated accessions. You see the red one is the wild one, the black one is the cultivated accessions of the Azerbaijan. We are going to publish this research. Hopefully, we have sent this research to very well-known journal. Hopefully, after maybe 15 days or 1 month, it will be online, and you will get more information about our fig project too. And also, I want to give you some information about our hazelnut genetic resources. We have started also this work within the PhD program, 𝗤𝗏𝗍𝗏 Askerova's PhD program. And first time we passportized all our hazelnut genetic resources. And now I want to inform you, it is the real number. We have 128 accessions of variety. We conserve all of them in this north part of Azerbaijan, in Zaqatala Experimental Station of Fruit Plant Institute. And then next slide, we started to study, of course, we did all phenotyping characterization, and we started also evaluation. First time we did molecular identification, just we used in the PhD program, we have used the ISSR markers, but we continue our research on the SSR markers too. And then first in our first experiments, we studied genetic diversity of several accessions, and our main purpose was to give the genetic passport to all our accessions. And next slides you will see we have the digital. It is our results. Then next slide you will see the grouping and the digital code. We gave the code to 12 accessions, but we continue all our experiments. Hopefully, we will give the genetic codes to 128 accessions. And next slide, I think you will see the digital code, and we gave such kind of digital code. It can be a digital identification code, or we say it is the digital passport of our accessions. But as I talked on figs and the pomegranates, we started also to study whole genome of the hazelnut. And in collaboration with China's Research Institute, we sequenced 100 accessions whole genome, and we identified almost 4 millions of them SMP markers. Please, next slide. 4 millions of them, we identified 4 millions of them SMPs. And we have also phenotyping data. That's why we are trying to do association mapping of some traits. We have some results, but we didn't publish. Now we are in the process of development of the new paper. Hopefully our paper will be ready within the 2 months, and then we will share all our hazelnut results in the near future. Dear colleagues, I would like to inform you, we study all the crops. We have a lot of research on the cereal plants, legume plants, technical plants. We also started to study genomic diversity of medicinal plants, and the fruit plants. We have data on the apple, pear, a lot of stone fruits, and etc. And here I would like to say we deeply want to collaborate with you on the genetics and genomics experiments. We have very good collaboration with the Turkish universities and research institutes, but we deeply want to collaborate with the Pakistani organization and other countries' organization. Thank you very much. I will be happy to answer your questions if you have. Thank you.
Thank you sincerely for sharing your innovative research and expertise. It was really nice seeing your research is so deep and so informative, filled with future innovations. Now we like to move for the question and session answer. If someone have any question, kindly write in the chat box. Due to the time shortage, we will move fast forward. Okay, I think no one has any questions. So thank you, sir, again for your amazing presentation, which gave us so much insight. And I think the participants got enough insights about their research and about future innovation.
Thank you very much.
You are welcome. So now we are honored to welcome Dr. 𝗔𝗴𝗿, Assistant Professor at the Department of Agronomy, University of Agriculture, Faisalabad, Pakistan. His research interests include... Yes, sir, you are audible.
Okay. Is previous research is done? Presentation is done?
Yes, sir, it is done.
Okay. Thank you so much, Dr. Abbas. Am I audible to you? Dr. Abbas? Hello, Dr. Abbas.
Hello. Yes, hello. I'm here.
Okay. Thank you so much, Dr. 𝗵𝗂 Abbas. Really insightful presentation. I really like it, and I learned a lot of things about your research in Azerbaijan. Nice to meet, and best regards, Ghulam Murtaza.
Thank you. I wish you all the best. Thank you.
Thank you so much. Have a good day.
To you too.
Yeah. So now, Miss Nopo, you can continue. Please invite the Dr. Shaista. Also, she can host this presentation.
Okay, sure. Okay, now we have to invite Dr. 𝗔𝗴𝗿, sir. Ma'am, Ms. Shasta, if you are audible, if you are present there, kindly take the floor and invite our respected speaker for the presentation.
Ma'am Shaista Shafiq is audible. Okay, she's facing some network problems, technical problems. So we will continue it. I would like to invite Professor Dr. Muhammad 𝗬𝗪𝗏 from Department of Agronomy, University of Agriculture, 𝗤𝗏𝗏. I would like to invite Sir floor for you, please. Thank you so much.
Professor Dr. 𝗔𝗴𝗿𝗁 sir, if you are available, kindly take the floor.
Hello.
Yes, you are audible, please.
So let me share my PPT from my computer. I have just seen the
Okay.
slide, so this is a little bit misintegrated. So let me share
Okay.
from my computer.
Yeah, sure, please.
Please let me know if my screen is visible.
Yes, your screen is visible.
Okay. Thank you so much, 𝗏𝗏𝗏, and thank you so much for all the presenters. I think I have learned a lot from their presentations, especially about the fruit and tea, because I am from agronomy, so we
Sorry, we can't hear you. Are you there?
Sorry, your network might be unstable. Kindly check, please.
I'm just wondering that during this internet connectivity issue, while they sort this thing out, I'm just wondering. I'm very much open to for collaborations, and
uh,
normally, sorry, if speaker is there, please continue.
yes, sure. So the climate change threatened food security and farm economics in vulnerable regions like Pakistan. So although the Pakistan contributes only less than 1% of the world greenhouse gas emissions, it is one of the 10th most vulnerable countries to climate change on the map of.
Sir, you are not audible again.
He's facing some network connectivity issues. Just wait 1 or 2 minutes. Yeah. Thank you so much, everyone, for your deep patience. I really appreciate it, and I hope your learning from these wonderful presentations. Yeah.
In the meantime, if any participants have any questions or any confusions, they can ask in the comment.
If you kindly allow what I was actually proposing earlier, am I
Yes,
allowed to?
sure,
Thank
sure,
you.
sure.
Thank you. I am head of Department of Biochemistry and Biotechnology at the University of Faisalabad, Pakistan. And my work stream is, our main research streams are, I'm working on wastewater treatment through nature-based solutions, nanobiotechnology, and through biochar. And second research interest where my postgraduate students work is we biosynthesis. We do biosynthesis of industrially and commercially important chemicals through agriculture waste material. So we use rice husk, corn cobs, sugar bagases. So all these agriculture waste material to prepare some important chemicals like biopolymers, epoxy resins, furfural, and some other chemicals. So I'm very much open for collaborations, and we are doing some work on poultry waste also in government projects at the moment. So and some collaboration we have developed with the international universities in Malaysia and lately in China also. So anyone who is really is working on those lines on sustainability or sustainable solutions for industrial wastewater treatment or agriculture waste material, turning them into some kind of useful products. And I would like to listen to your work and to maybe work in collaboration. Thank you.
Sure, ma'am. That was really nice hearing from your research and your work. To be honest, I'm also from biochemistry and biotechnology, and I'm really surprised seeing the year or the era is really improving. I would really request if anyone has interest on that regard, kindly contact with our ma'am. Now, we would like to invite our next guest speaker. We are honored to invite Dr. Mujahid Hussain, Research Associate at the Department of Plant Sciences, University of Manitoba, Canada. His research focuses on precision agriculture, UVA technologies, and crop productivity enhancement. His presentation topic is optimizing UVA-based spraying, droplet deposition, and efficiency of 28 homobracinolide in wheat. Sir, the floor is yours. Kindly proceed with the presentation, please.
Okay. I'm trying to share my screen. First of all, I want to check if my audio is working. Can you hear me all?
Yes, your audio is visible. I think your presentation slide is also showing here, so you can proceed with that.
Right. I will share my own screen. I'm trying to share my own screen.
Okay, sure. Take your time.
Oh yeah, this one. Okay, hopefully you can see my screen now.
Yes, it is visible.
Yeah. So my name is 𝗤𝗂𝗿𝗄𝗃. Right now I'm working as a research associate at the Department of Plant Science, Faculty of Agriculture and Food Sciences, University of Manitoba in Canada. This is in Manitoba province. I just recently joined this institute a year ago. Before that, I have been working in China as a postdoctoral fellow in Shandong University of Technology. So this presentation is based on the research trial that I'm sharing right now. This is already published in journal 𝗔𝗴𝗿𝗀𝗃𝗿 as we're a publisher. So I will not go deep down, and I will not show the vast topic related to the agriculture, as I am an agronomist, and right now I am working on the drone and remote sensing direction in an agronomy field. For now, I will only present the small part of the remote sensing, like I will only discuss an inside topic about the spray drones. Although remote sensing control contains a lot of other topics that today I will not go deep down, as I said. Maybe in later incoming seminars or webinars, I will share my research and my experience and my trials related to the remote sensing topic. But today I will go through my spray drone experiment that I conducted with our with our collaboration with Chinese University. Today, my topic is optimizing UAV-based spraying and droplet deposition and efficiency of 28-homobrasinolyde in wheat crop. Homobrasinolyde is a growth regulator that is applied in different crops to increase the yield and senescence of the different kind of crops like maize and wheat. Let's start with the contents of this presentation. First of all, we'll go through with the introduction and background and objectives of this trial. And then I will go through with the sampling environment method that I have been using during this trial and different analysis. And then I will go through with the results that I analyzed and the conclusion, and then featured prospects and recommendations for the farmers. In the current cereal situation in the world is that the maize, I have conducted this experiment on the maize and wheat both, but today I will share only the wheat insights. So the cultivation area for the maize it's about worldwide is 197 million hectares under cultivated maize crop, and 216 million hectares are under the cultivation of wheat, which shares almost 39 and 26% of the world cereals, and the total production of the maize and wheat worldwide is 1,137, while for the wheat is 757 million metric tons per year, which is 27% of the world cereal production as a maize, and the wheat shares 30% of the world cereal productions worldwide. So the main problem is the climate change. Yes, this is real. Few years ago, this topic was discussed in many countries. Is that real or not? So now we know the current situation of the global weather like in different countries, we can see the global weather situation, so we can know that the climate change is real. So in this current climate scenarios, it is expected the increase in the 90.6 billion population until 2050, according to the estimates. So where will the food come from? This is the main concern. This is our main concern. The food security is the main concern, which is really affected by the biotic and abiotic factors, which are included the insects, pests, and also included the rainfall frequencies, like we can see in recent years, and the rainfall disasters, cloud bursts, then how to deal with these abiotic factors. So, and the rising temperature, yes, this is also very serious, unexpected weather changes, and which are very important for the agriculture productions. So what do we do? What should we do in this situation? There are tons of other methods that we can use, but today I will not go through with all. I have selected only the precision farming, and also the use of plant growth regulators and its application methods. How efficiently should we apply the growth regulators or spray these regulators, the efficient use of these chemicals, for which is very important concerns for the farmers, and how precisely can we apply these chemicals on the different crop without affecting the environment and without affecting the farmers' pockets. So today I will go through with this strategy to apply these growth regulators with the different sprayers onto the wheat crop. And also I will see the different effects of these plant growth regulators on the wheat crop production. The next segment is the sampling environment that I used during my experiment. In this experiment, the different two kinds of sprayers were used. The first one is T50 DJI drone. This is the sprayer drone. Now the T100 is released, but this experiment was done a few years ago, and this experiment is based on 2 years of repetition. And that time we don't have a T100. Now this in 2026, I think T100 is released, and we don't really need it because this T100 is used in the vast fields. This is not very good for our small experimental trials. So we use T50 sprayer, and with this we needed to compare the another drone. There are other drones like boom sprayers and other lot of sprayers, but here we only use the manual knapsack manual sprayer, which is Supergreen 60 Matabi company from Spain. And in this experiment, to clock the droplets, we use the different two approaches, which is a droplet collection card. This is a very important approach that every researcher used to collect the droplet cards. Let me open the laser. Okay, I don't know if I can open it. Okay. Yeah, here, like I said, I use these droplet collection cards on the both maize and wheat. Today I will go through with the wheat. And these droplet cards are weather sensitive papers with the different dimensions, that one was put on the wheat head, another was on the flag leaf, and other was put on the bottom leaf of the wheat. So this is the experimental layout, and these pictures shows the different water levels that was used in the drone, like 15 liters per hectare. The another water level was 30 liters per hectares. These are the droplets collection cards which I put on the wheat, and after sprayers we waited for the 30 minutes and then remove these cards. And this is the final loc of these scars, which has collected the different droplets, spray droplets. Like this is the low volume, this is the high volume, spray volume I'm talking about, and the different dosages and the same nozzles, the general nozzles that are being used in the T50 DJI drone. After collection of these cards, this is the single card which was scanned at 600 dpi with the scanner, and then it was gone through with the 𝗦𝗂𝗀𝗃 software. Yeah, this card was inserted into the 𝗦𝗂𝗃 software to collect the droplet coverage, droplet density, droplet deposition, and droplet deposition rate. Other method also was used to check the droplet deposition. The leaves were taken from the plants and those were washed away in a constant water level. And this water level was collected into these small bottles, which were going through with the spectrophotometers. And then with this method, we measured our droplet deposition and droplet deposition rates. Other parameters were taken during this experiment, like photosynthetic parameters. We used IRGA, which is infrared gases analyzer, made in USA. This was used to measure the photosynthetic parameters. Photosynthetic parameters, this can measure a lot of photosynthetic, like I think 16 to 20 parameters, but only we have taken 4 kind of parameters, including internal carbon dioxide, which is called, and net photosynthetic rate. With this, we also took internal transpiration rate and stomatal conductance. These 4 parameters we took from this instrument, which we will see the different results in the next incoming slides. These were the main objectives of this study, to compare the possible effects of 𝗔𝗴𝗃𝗃 on the physiology of wheat crop applied by the UAV and knapsack manual sprayer. Another objective was to evaluate the droplets deposition spread by the knapsack manual sprayer and the spray volumes. 2 spray volumes were used, like 15 and 30 liters per hectare by the UAV and on the different plant canopies, including the top, middle, and the bottom of the plant, by selecting the different leaves from the plant. And the third objective was to examine whether the results of the knapsack manual spray and the UAV are different when the 𝗔𝗴𝗃𝗃𝗃 dosage were decreased by 20% in UAV compared to the knapsack manual spray, because we also used the 𝗔𝗴𝗃𝗃𝗃 different dosages in this experiment as a factors B. Now we will go through with the results that we obtained with the evolution of droplet deposition and efficiency of 20 𝗔𝗴𝗃𝗃𝗃 spray, the different sprayers on the wheat. Here is the experimental treatment plan. The UAV 1, 2, 3, 4, 5, 6 KMS and CK was used to compare all these treatments, and the knapsack manual sprayer, only single treatment with the 24 milligram active ingredients per hectare was used for the comparison, and 450 liters per hectare spray volume. This is the water level was used, and we did not use any adjuvant here to spray by the knapsack manual sprayer. These adjuvants were used normally for the droplet integration on the plant leaves for only drone application, not for the knapsack manual sprayer. And the UAV 1 and 2 contain the same dosage of the chemical, but the 2 different kinds of water volumes, like 15 and 30 liters per hectare, 3 and 4 for 15 and 30 with the middle dosage, and the 30 liter per hectare for the highest dosage we used for the 𝗔𝗴𝗃𝗃𝗃. These are the weather conditions, as this experiment was based on the 2 years of repetition, the first year weather condition and the second year weather conditions during the plant growth month that is started from October to the June because the wheat was sown in October and harvested in the next year, June, and that same experiment was repeated on the next year, October 2024, and it was harvested in June 25. This is the weather situation. This is the mean temperature that was noted highest in April, and this was just not as highest as the second year. So we will see in the results this temperature has a significant impact on the different treatments of our experiment. And this is the precipitation graph and comparison of precipitation and the different temperature. So this is gaseous exchange parameters that were taken from the 𝗔𝗴𝗿. So we got net photosynthetic rates and the stomatal conductance from the 𝗔𝗴𝗿, and the two other parameters like the stomatal conductance and the internal carbon dioxide also we took, and which we'll discuss later. We see the treatment effects. So we got the significant treatment effects in the first year and also in the second year. So these higher treatments are the middle dosage and the highest dosage of the chemical, and these two are the 15 and 30 liters per hectare. Here we can see the 15 liter per hectare is applied by the drone, and 30 liter per hectare applied by the drone are higher as compared to the CK and the manual sprayer. Here these are the lower dosages that has no effect, and there is no effect between the water levels. And this trend followed the same treatmental effects in all the parameters that were taken by 𝗔𝗴𝗿, like net photosynthetic rate and stomatal conductance. Here we can see the same behavior. These data were taken after every 2 weeks, 15 days after the application of sprayer, and the 30 days after treatment applications. If we see the intercellular carbon dioxide in the wheat and the transpiration rate, this also showed the same behavior with the treatmental and water level effects on the crop plants. One thing I want to mention is that we applied this chemical on the flowering stage of the wheat crop just before the pollination. We have a look on the SPAD chlorophyll of plants, and these are the different treatments and the different sprayers, different water levels that were taken on every 10 days interval. Here we can see on the 0 day, of course, the treatment has no effects on the plants, and the treatmental effects were significant on the 10 and 20 days after the application of these chemicals with the different sprayers. Here we can see the different significant effects between the factor A, factor B, like between the different sprayers and also the different chemicals and the spray volumes. Almost spray volume are similar within the drone, like 15 and 30 liter. 30 liter has less chlorophyll compared to the 15 liter. This is because the droplet penetration is better in 15 liter per hectare compared to 30 liter per hectare. And also here the second year has the same behavior like the first year. If we talk about the droplet deposition that we measured using the ImageJ and that droplet collection cards, there are 4 different kinds of parameters that were collected. The first is droplet coverage percentage, another droplet deposition density, droplet deposition and rate. So first we understand the droplet coverage and the droplet density idea. What's the difference between these two? Droplet coverage is how much percent of these droplet cover the droplet connection card, that how much area, how much percent was covered with the droplets. And this is droplet deposition density, means how many, like these are the numbers, how many droplets were there on the droplet collection card, how many per centimeter square. So this is different between these two, and this was collected by washing off the plant leaves and then gone through with the spectrophotometer to collect the droplet deposition microliter per centimeter square, which was measured with the 700 wavelength, and this is droplet collection rate. So here we can see on the wheat head, wheat flag leaf, bottom leaf, and this is the average. If we will not talk about the head and different parts of the crop, we'll only see the wheat head has the most droplet coverage percentage compared to the flag leaf and average. So the wheat head got more coverage of the droplets compared to other parts of the plants. And if we talk about the droplet density, also, of course, the droplet density is also higher with the 15 liter per hectare. And this is the 15 liter per hectare, and 30 EAP has most droplet density compared to the 15 and 30 liter per hectare of the drone. Of course, the 450 liter per hectare, if we compare it with the 15 and 30 liter per hectare, this is the most. But the main important of our concern is the droplet deposition. How much droplet deposition with the drone, the UAV, with the 15 and 30 liter per hectares, and the knapsack manual sprayer. So knapsack manual sprayer was the recorded lowest deposition, and higher deposition was found in the 15 liter per hectare compared to the 30. In all the different parts of the plant, we can see the 30 and the 15 liter are higher compared to the knapsack manual sprayer. So we can say the 15 liter per hectare with the normal dosage performed well according to the droplets data. Now we'll go through with the different yield parameters. We will see what happened with the yield of the crop. Okay, before going to the yield, we will see how accurate were these sprayers performed in the field. If we see the CV, so the CV was lower in KMS because of higher water levels, and the CV between the 15 and 30 liter was higher in the drone. As because the water sprayer level in the drone is lower as compared to the knapsack manual sprayer, so the CV is higher. This is the main reason. So that means the spray done by the knapsack manual sprayer was more uniform compared to the drone. But we shall see the results that how higher was the yield was affected by the KMS and the knapsack manual sprayer and the UAV different water levels. The grain filling dynamics, we also measured the grain filling dynamics in this crop. That was measured in every week after the application of these chemicals. We can see the different treatments, the grain increased, like in the 7, 14, 15. The highest grain weight was measured as 35 days after the application of these chemicals. Then this grain weight was constant, and then after 42 days it was constant. Almost after 42 days the maturity and plant go to the physiological maturity, and also this goes to the harvesting stage. So this is the first year and this is the second year performance. If we go to the HBR and spray volume effect on the final wheat grain yield, the wheat grain was divided into the different sections. The first, it was the main concern was the grain, and this was divided into the spike and bloom, and the third part was the leaf and stem shape. If we can see here, the UAV3, the UAV3 means the 30 liters per hectare with a middle dosage performed better compared to the every other treatments, like 15 liters per hectare with the middle dosage. This is also same, no significant effects were found between the low dosage 30 liter per hectare and 40 liter per hectare. If we talk about the low dosage and medium and high dosages, the medium and high dosages were almost same compared to the low and the control and knapsack manual sprayers. This was all the same in the different parts of the wheat. And if we see here, almost similar differences were found after doing the statistical analysis. If we see only the homobraucinolide and if you see the spray volume, we can also see the big difference between these different treatments, like the 19 dosage and the middle and high dose. Yeah, between the 𝗲𝗿𝗏𝗃 and light dosages, that significant results were found. Now we'll go to the conclusion without wasting time. We concluded that the dose of 𝗲𝗿𝗏 between 24 to 30 mg active ingredient per hectare was performing well. This is the range between medium to high. So there is no use to use higher dosage. So if the higher and the middle dosage is performing equally, then of course we will go through with the medium dose instead of using more chemicals, which cost more to the farmers. And for the farmers, we recommended 24 to 30 mg of 𝗲𝗿𝗏𝗃 at the flowering stage of the wheat, sprayed by 15 liters per hectare by the drone, which is used for the medium range farmers, not for the vast lands. For the vast lands, the 300 is recommended. For the medium range farmers, farmers can use T50 drone with a 24 to 30 mg per hectare with a 15 liters per hectare spray volume was recommended for the farmers. So we do recommend some corrections and some experiments which are needed to conduct in the near future, because we found some clause in this experiment that UAV has a less uniformity as compared to the knapsack manual sprayer. Now the drone companies are working on the different nozzles that are more efficient compared to the different sprayers and other drone company nozzles. So they are paying more attention to modify the nozzles, which can be used to uniformly application of different chemicals into the different crops. And we also think that further study should be conducted for the comparison of other brasinolide and phytosteroids, which is an important part of the green technology, which can be applied by the UAV and other conventional sprayers on the different other crops. Like we have already done this on the wheat and maize, and we are planning to apply these on the different crops. And also we are planning to extend these approaches in the Canadian prairies, because in China, the application of pesticides and these chemicals are allowed to apply by the drone. But here in Canada, it is not allowed to spray the chemicals by the drones, although they are spraying chemicals using the tractor drone sprayers. Sorry. And other sprayers, but it is not allowed to apply these spray chemicals by the drones. But hopefully in the future, we will get permission from the government, Canadian government, to allow us to apply not the hazardous chemicals, but at least we can apply the growth regulators, which are not very harmful to the environment and other species like insects and plant species. At least we can apply the plant growth regulators, and at least we can apply these liquid fertilizers that are used to be sprayed by the drone with the nozzles and spray conditions. So another recommendation is this is for the companies to increase the power storage of the flight, because this drone flight can only go up to 10 to 15 minutes, which is not favorable for the big areas or for the big scale farmers. This is not suitable for that. Thank you very much for your attention. If you have any question concerning with these applications, the crops related to agronomy, you are very welcome.
Thank you so much, sir, for your such an amazing and informative presentation, from the spraying equipment to the droplet deposition, and lastly photosynthetic parameters as well. It was really insightful. Now, I would like to invite the participants. If you have any questions, kindly write in the chat box. Okay, I think our
I
participants.
believe I have cleared out everything. Nobody has questions.
Yes, sir. Your innovative research and expertise was amazing, and our participants do not have any questions. Thank you again for your time, appreciation, as well as your consideration. That was a nice presentation you delivered to us.
Thank you very much. You too.
Mr. Murtazak, if you want to take the floor, kindly take the floor.
Yeah, sure. Thank you so much, respected sir, Dr. 𝗤𝗂𝗍𝗍, for wonderful presentation. I really learn a lot from it. Moving forward to it, I think there is no question from. If anyone have any question, you can ask in the chat box. We will, yeah. And right now, we will conclude over this session. Along with us right now, there are our international speakers available, Dr. 𝗂𝗍𝗵𝗏 and Dr. 𝗤𝗂𝗍𝗍, Dr. Kanal, and yeah, I really appreciate to be stay connected till the end of all presentation, especially to the Dr. Kanal and Dr. 𝗂𝗍𝗵𝗏 and Professor Dr. 𝗤𝗂𝗍𝗍. So as earlier we just discussed in our pre-planning meeting to open the new doors for the collaborations along with 𝗬𝗏𝗏 International Alliance for Sciences, where this alliance have the partnership in all about 30 countries in all over the world, in Asia, in Europe side, and yeah. So we are going to move on towards the launching of international journal. Our software engineering teams and data analysis team are working behind the scene. So soon we will launch an international journal in the domain of plant sciences, which have the open access for the young researchers in all over the world to promote science and sustainability more. So I would like to invite our respected guest speakers to give the closing remarks, and then we will conclude it. Thank you so much, everybody. Thank you so much, everyone, to listen me. Best regards, 𝗤𝗏𝗏 Murtaza, CEO and President, 𝗬𝗏𝗏 International Alliance for Sciences. Nupur, please invite one by one.
Sure. First of all, Ma'am 𝗂𝗍𝗵𝗏, kindly take the floor and share your closing remarks. Okay, ma'am shared in the chat, which is I would like to invite Sir Mujahid Hussain to give the closing remarks.
Yeah, thank you very much to all of you for sharing the insightful topics and your presentations. It was great to hear from all of you. It was very knowledgeable. And hopefully I have one recommendation and also one request to the organizers. If we are arranging the next any webinar or any conference, it's my humble request to ask the speakers to follow the time that they are allotted according to the schedule. This is my humble request to the organizers. Yeah, because here I have seen that we have not been following the time that were allotted in the announcement, right? So please try to follow the timetable. And yeah, thank you very much all for being here, for sharing your ideas. And one more thing I want to say, that if anyone is interested to collaborate with us, we have our website with 𝗔𝗴𝗿𝗀𝗔𝗴𝗿.ca. You can just go, you can contact us, and you can share your interest if you want to collaborate with us in remote sensing, in weed science, and any field of the weeds in different agronomic crops. So you're most welcome to collaborate with us and visit our website. Thank you very much.
Thank you, sir, as well for your suggestions and your future collaboration. Now, I would like to invite Ma'am Kunal. If you were present, kindly give your closing remarks. Okay, I think Ma'am Kunal is facing some networking issues. As we are in the end of the conference, I would like to sincerely express my heartfelt gratitude to all respected speakers, organizing committee members, collaborating institutions, and participants, and the attendees from around the world. A special thanks to all moderators who work behind the stage, technical teams for ensuring smooth engagement and this international event. We hope this Congress becomes a beginning of many future collaboration, impactful research initiatives, as well as scientific partnership across borders. Thank you everyone for your valuable time, presence, and contribution. Wishing you continued success, health, and excellence in your scientific journey. Stay connected and stay inspired. Allah Hafiz. Goodbye to everyone.
Thank you so much, Mr. Proctor, as well for making that uh, international congress in a very meaningful way. I really appreciate your efforts. I hope my message. Hello, Nurpo, am I audible? Nurpo, am I audible?
Your voice is low.
Okay, is now I hope audible. Yeah.
Yes.
Okay. Thank you so much, Dr. Mujahid Hussain, Dr. Kenol, and Dr. Keziban. I really appreciate your efforts and your contribution in the first International Congress in collaboration with the cross-border countries, Pakistan, Turkey, Azerbaijan, and Nigeria, along with Canadian some scientists. And I also appreciate the efforts of our respected participants. Thank you so much for your patience. I hope you will learn a lot of things and be connected within the group, so we will make
future collaborations in the domain of plant sciences, integrated plant sciences and life sciences. Like you can see the Dr. Salio presentation, which was on phytopharmacology. So it's, how can we say, like plant sciences integrating almost with each domain of life sciences. So looking forward to it. I would like to request all of you, stay connected within the group, be active, stay focused, and wish you all the best. And thank you so much again and again. And Allah Hafiz. Right now we have to conclude it. Miss Kanol, Dr. Kanol, if you listen me, please. Dr. Kanol. Is Dr. Kanol here? Okay.
I think she's facing some network issues as well.
Okay. So we have to right now conclude our this journey. Thank you so much, dear participants, for your meaningful contributions in engagement. I really like your passion for learning, especially in the domain of integrated life sciences. I hope you learn about for the best, and wish you good luck. Now, Doctor, you can now conclude it.
Okay. Thank you, everyone who joined with us, and thank you for your patience as well. We really appreciate your time, as well as our respected guest speakers for presenting their slides that professionally. Thank you, everyone, and see you next time. Allah Hafiz, everyone.